remarkable event, evolution might have become stuck at this point. Single cells, or thin sheets of identical cells, could control the contents of their internal water, as long as they remained immersed in the constant environment provided by the sea. Any cells that were not directly bathed by seawater would have been in danger of drying out. What made the next step in the evolution of more complex forms of life possible was the appearance of special organs to monitor changes, such as drying out, and to link these to some mechanism that would allow it to protect itself. This required a communication system to be developed. The various steps in this evolutionary pattern are speculative, but one can make a reasonable guess as to how they came about. Some indication is afforded by studying embryonic development and the stages that animals, such as the frog, go through in their maturation process. It starts with a single fertilized cell, the ovum, and develops into an embryo containing many apparently identical cells, into a stage at which cells with differing functions start to appear. This allows its maturation into a fish-like tadpole, which lives in water, and eventually emerges from its watery environment as a young, air-breathing frog. It is probable that the only form of life that was capable of leaving the primordial swamp and surviving on land was composed of one or more simple, identical cells. This form of life would have survived by absorbing water and dissolved nutriment through the thin membrane covering the cell. The main danger to its survival would have come from changes in temperature or from a drying out of the environment. This dehydrating effect would have produced changes in the local concentrations of chemicals, such as sodium, potassium, chlorine, and calcium within the cell. It is believed that it was these alterations in the local concentration of salts that initiated the process of differentiation, in which new types of cells were formed. It was this process that led to the development of more complex organisms. With the passage of many millions of years, the atmosphere became drier, and temperature fluctuations more dramatic. Only those organisms that produced some means of adapting to these changes survived. Those that survived developed groups of cells that had special functions. These were the forerunners of the sensory and motor organs found in more advanced forms of life. Because of the drier environment in which they now existed, the surface of these early animals became covered with a thick skin, or carapace, to limit fluid loss. These impermeable covering would have prevented the organism from taking in fluid and nutriment through its surface, and necessitated the development of special mouth areas through which food and water could be ingested. As the environment dried out further, and changes in the temperature became more extreme, the chances of survival depended on developing some means of locomotion to allow it to escape from a dry, hot area for a nearby site where water, nutriment, and shade were available. This required some means of detecting changes in the environment, a sensory system, and a means of conveying the information from these sensory organs to the organs responsible for propulsion. For this to be possible, a messenger system had to be developed. Chemicals, acting as messengers, served this function. The first such chemical messenger must have been formed from substances that were abundant in Earth's atmosphere at that time, such as ammonia, carbon dioxide, and water. It is probable that this was either acetylcholine or a similar substance. It would have been released in response to an alarm signal from cells capable of sensing changes in the outside world. It is quite possible that the present-day role of acetylcholine, the most ubiquitous of all the chemical transmitters in the animal kingdom, dates back to these primitive times. It would have functioned in much the same way as it does today in the clams and oysters on our seashores. In these bivalve mollusks, acetylcholine is released into the fluid, bathing the tissues in response to a signal produced by touching the shell. It causes the strong muscle that closes the two halves of the shell to contract. Even today, an atavistic enzyme that destroys acetylcholine is present in the bloodstream of man. No physiological function can be ascribed to this enzyme. Indeed, a number of people live perfectly normal lives without it. It is possible that its presence is a footprint of the evolutionary stage when acetylcholine was released directly into the tissue fluids. As evolution progressed and these organisms became more complex, special structures capable of detecting changes in the outside world, such as eyes and ears, developed in parts of the body that were remote from the muscles involved in making an appropriate response. This necessitated a more sophisticated